Okay, thank you. Thanks for the update. Yes, so we were talking about two levels. First is at the information level about the we are learning about how genome is organized. The first one is about the information, unique sequences, and also about the repetitive sequences, intergeneric sequences, and so on. That is at the level of information. The second way of looking at is how is the genome physically organized, physical organization of the genome itself. What kind of proteins, the supercoiling, and many other stuff like that. So we also had a discussion about nucleosomes. What are nucleosomes and amount of DNA that is wound around each nucleosome. Nucleosome consists of histones, and histones can be, there are 2 or 3 different types of histones. Let me erase a few things so that we will discuss in this place. Yeah. So there are 2 types of histones. There are core histones and there is linker histone. In the core histones, we have H2A, H2B, H3, and H4. As H2A and H2B occur as a dimer. H3 and H4 occur as a tetramer. And linker DNA is, linker histone is H1. And that is the composition of one nucleosome is about, it has total of 8 histones, 2 of each, 2H2A, 2H2B, 2H3, and 2H4. But they are bound as H2A and H2B bind as dimer. When I say dimer and about nucleosome, when we are talking, it is about 2 dimers and one H3H4 tetramer, right? And that is how the nucleosome is organized. We also learned about there is between nucleosomes, right? This is a nucleosome, this is another one, and so on. There is gap. Say, for example, this part is the linker DNA between 2 nucleosomes. Okay, so the spacing or the spacing between nucleosome could vary within a species and among different species as well. The amount of DNA is usually about 147. There could be here and there little bit changes, and it binds around 1.6 times. That means this is 1 time, and this is about 1.6 times, something like that. And here would be the core histones or the nucleosome, right? And the outside ring that I have shown is the DNA wound around it. So in today's class, we will see a little bit of higher organization. That is a important question that usually comes up in examinations. We haven't spoken about H1 and what is the function of it in general, not so much, but we will do it. So the around DNA winds around the histones about 147 base pairs. And the H1 histone that binds to the linker DNA, the region outside here somewhere, it binds somewhere here and then makes much more tighter complex. And because of which, say if H1 is present along with the core histones, it protects about, I mean, it is, you can say something like 167 base pairs are wound around the DNA. If there is no H1, typically you will find only 147 base pairs are wound around DNA or the nucleosomes. Okay. So the function of H1 is to bind to the linker DNA in between 2 nucleosomes, and then it will make it much more compact. We'll see in the other slides. So here is a nice picture of depicting the effect of H1 histone. This is a nucleosome 1, 2, 3, and 4. Okay, and they are actually much more dispersed. They're connected, but they are dispersed. If H1 histone is wound, then it becomes much more compact. You can see the amount of space or the length of the DNA that is here And now upon binding of H1, you have it almost 50% is occupying the lengthwise. It is actually only 50%, right? That is the effect of H1. It compacts DNA further. There are the next level of organization. First, we will see the overview also. First, we have learned about double-stranded DNA. Then we learned about nucleosome formation. And then we are going to learn about something called as 30 nanometer fiber, right? That means how is DNA much more compact. You can see that it is almost like a spring loading onto one and becoming much shorter. So there are two possible ways of DNA winding around itself. One is called as solenoid, and the other one is called as zigzag. If you look from this view, if you're looking inside, you would look. It would look something like this. In solenoid, the arrangement is something like spring actually, right? And then you'll have higher levels and so on. So you can actually, if somebody is holding at this end and someone else is holding here, and you can stretch it, it will come out. It is much more prone to opening compared to something like zigzag. Zigzag is like this, it's going, and then you can see that there is a lot of intertwining. And if one tends to pull, sometimes maybe it's not as open as something like in the case of solenoid. Okay, so different parts of the genome could be in different ways. It depends on the expression activities and so on. These are some models. I think mostly it is about the solenoid model is thought to be the better explanation. So what stabilizes the 30 nanometer fiber by histone tails? It is by the histone tails. Before we go, we will have a quick. So 30 nanometers, it's called from here till here. If you look, that is what the length, the diameter of the fiber is, 30 nanometers. Therefore, you call it as 30 nanometer fiber. So here are, I hope you realize that this is the zigzag one, right? It is going to one to the other side and so on. If we recollect in the last class, this is the core histone and their tails. Each of them we were discussing also that is the end terminus of the histones are usually projecting outwards, like as shown here. Those are referred to as histone tails, and they have important epigenetic regulation in epigenetic regulation that we will discuss briefly in the next classes or next part of the lecture. So these histone tails have they can make a lot of hydrogen bonds with other histone tails, and because of which the 30 nanometer fiber will retain its shape like this. Okay, stabilization of 30 nanometer fiber by histone tails. How do they do it? By using hydrogen bonds between tails of adjacent nucleosomes. So this is kind of overview of how the organization is present. This is the DNA wound around nucleosomes. It is about 10 nanometers, and then it can form a zigzag or solenoid that is about 30 nanometer fiber. This 30 nanometer fiber can form a loop like this, okay? And there are multiple loops like this. And then you see one chromatin fiber. This is called as one chromatin fiber. There is a chromosome scaffold onto which there are several loops. One DNA loop is forming here. Each loop consists of it's a loop of 30 nanometer fiber. Each 30 nanometer fiber is made up of multiple nucleosomes, and nucleosomes constitute naked DNA and, of course, histones, right? And during metaphase, you will have even more complicated or complex organization. So while we discussed about different types of histones, core histones especially, like H1, H2A, H2B, H3, and H4, There are also variations in histones itself. There are different types of histones that are being produced. One is called as H2AZ. It is a variant which is involved or which gets incorporated in place of H2A. From the name itself, you can identify that, right? They are usually associated with transcribed regions. So when we are reading about regulation, gene regulation, what you should, or for the moment, what I want you to remember is something like this, right? In one of the explanations or trying to put the importance of it, as I was saying, the genome in all of our cells is same, almost identical. But still we have very different cell structure cells in our bodies. How is that happening? That is primarily because of differential gene regulation. Gene A, B, C, and D, say these are all different genes. In some cases, in some cells, some genes might be on and others might be off. In some others, A and B could be off and C and D could be on. So what I'm trying to say is not all the genes of genome are expressed in every cell. There are some housekeeping genes that are required for metabolism, replication, transcription, translation, and other things. Those will be on all the time. Those are called as housekeeping genes. Then there are also genes that are regulated, right? Which means they can be suppressed or expressed based on need. And there are also tissue-specific genes or cell-specific genes. Say in neurons, you need to form a cell, and in that case, only neuron-specific genes will be expressed. In no other cells will those genes be expressed. So what I want you to remember is some genes are highly expressed, as in the case here, and some genes are highly repressed. So here, H2AZ is a variant of H2A, which is usually associated with highly transcribed regions, right? And because of their presence, they will inhibit the formation of repressive structures. You don't have to remember or memorize all of these. You will, as we progress in the course, you will understand when it comes to gene regulation. You will come to that level, and then we'll be able to understand it better. Then there is another one called as CENPA, which is a variant of H3, which also functions something similar, but it is organized near the kinetochore. Or kinetochore is something that organizes near the centromere, right? Say this is the centromere. This is where kinetochore is organized on top of the centromere, to which the spindle fibers bind like this, right? So these tails of CE and PA are good. Those are good to, that is where the kinetochore protein will bind and form the kinetochores. Otherwise, kinetochores can form anywhere else, right? But this is CNPA is the protein which is a variant of H3A histone, which will determine the binding of kinetochore protein, and these are usually associated with centromeres. And what is the function of centromere? It is important or essential for appropriate and accurate segregation of chromosomes during cell division. If a chromosome has more than or less than one centromere, there will be problems with segregation. If there are less number, the daughter cells may not receive appropriate number or may receive more. If there are more than one centromere in a chromosome, it might result in fragmentation of a chromosome. So we will quickly have a brief discussion on regulation of chromatin structure, and let's see how, as I said, the chromosome, if the DNA is wound around and formed histones, and then we have 30 nanometer fibers, DNA loop, and so on. If that is the case, how are genes expressed? Right? Genes must be expressed, and to be expressed, if it is in constantly in the same structure as nucleosomes like this, then it won't be, it's not so um, favorable for gene expression, because gene expression requires not only dissociation from the nucleosomes, but even the DNA itself should open up something like this. Denaturation is required, onto which RNA polymerase may bind and produce a mRNA or RNA itself, right? So therefore, the 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 organization should be much more dynamic. It should be vulnerable. It should be able to open up and close up right after transcription is done. So we'll have a quick discussion on that now. Yeah, this is where I was talking. The interactions or the organization should be dynamic, because transcription should happen and also replication should happen. And all these, majority of the interactions that you see here are usually non-covalent interactions. And majority of them would have a lot of hydrogen bonding, like we have seen about the histone tails extending outside and then interacting with other histone tails using hydrogen bonds and so on. There are also covalent interactions, such as modifications of histones that we will see in a later slides. So here is how can a protein become a piece of DNA or sequence of DNA become accessible to the proteins, interacting proteins. Here is an example of two protein binding sites. Here is site 1 and site 2, indicated blue and red. And if it is at the edge as shown here, then it is a frequent event, because it is the nucleosome could slide backwards, and in that time this DNA can get exposed, and then protein 1 may bind here, and that's usually a frequent event, because these are not completely or rigid structures. They are much more flexible, or the nucleosome can slide up and down a little bit. The blue one, the protein binding site 2, is much more interior in the nucleosome formation here, right? Very rarely though, it's a rare event that so much of DNA could open up and protein 2 could bind there. But it is a possibility. That is how the nucleosome, that is how the DNA binding proteins can bind to their respective sites, and depending on the position of these respective respective binding sites, they will be either frequent or rare events. So, but if they are somewhere, something like shown inside in here in this one, or if it is here, they are much more difficult to be accessible. To do those things, to facilitate interaction, there are complexes called as nucleosome remodeling complexes. They consist of multiple proteins. They form into one complex, and then they can change. They change a lot of things, which we will see in the next slide. But first thing, they can change the nucleosome position, interactions, and they can interact with DNA, and they require ATP to facilitate the remodeling process itself. You don't have to remember these names as such, sliding, transfer, and remodeling, because I think in the new edition, there are new terms. We will follow those. In the older one, it used to be sliding, transfer, and remodeling, but that's all right. Here are sliding. There are three models, remodeling possibilities. One is sliding, the other one is called as ejection, and the third one is called as dimer exchange. So these are three remodeling complexes. And these are kind of models. Some people propose some, and possibility is that the three might be there. And with more knowledge in the future, probably these will be modified as well. But for the moment, we will understand it this way. I hope you see that there are 1, 2, 3, 4, 5 nucleosomes, and there is a binding site here. And if nucleosome 3, right, it slides this way, and all these 3 nucleosomes slide the opposite way, that will expose the binding site here, and then the protein can bind here, and then make an interaction and do whatever it is doing, either expression, repression, or any of those. There is something called as exchange, ejection. We used to call that in the previous ones as transfer, where the whole nucleosome 3 is ejected out. Nucleosome 3 is ejected, so that now this whole DNA is accessible by the binding proteins. There is another way. It is called as dimer exchange. So H2A, when I say dimer, it should be a H2A H2B dimer, right? If I want to say something else, I should have said as H3. If I said tetramer exchange, it could have been then H3 and H4, right? Because it is said as dimer. What is most likely exchanging is H2A, H2B dimer is being exchanged, because it could be a variant, because of which there could be small changes in the, that is one another remodeling. Because of the variant addition here, new things may happen. Okay. So there are several proteins, complexes, SWI, SNF, ISWI, CHT, INO, AT, and so on. We don't have to memorize or mug up all these things, but you need to remember a few things here, as what is a bromodomain and what is a chromodomain. As we go a little further, you will see that. What can they do and how can they do it is whether sliding can be done by these complexes or not. When you say slide, it means the nucleosome can be slided or not, like we have seen in the previous slide. And whether transfer can happen. They can do transfer. Transfer is something like ejection from one DNA to that nucleosome is removed, and probably it could be bound by DNA from a different place, but that's all right. Just remember bromodomain and chromodomain. When we get to the later part, it will be useful, or probably in the next few slides. So first of all, if we have DNA, say this is the DNA molecule, we have a DNA like this, and say it is a double-stranded DNA. Where should the nucleosome form? Where should it not form? And how these things can be determined. So it is interaction of histones with DNA is non-specific, right? It binds to DNA irrespective of what the sequence is. But there are some places where nucleosome form in abundance, and in some places where they do not form in abundance, or they form very weakly, right? That could be directed by positioning. Positioning could be directed by DNA sequences. I will tell you, it is not sequence specific, but it's about whether it is AT rich or GC rich, like that, like general terms like that. Or it could be by some other DNA binding proteins, which will help the nucleosome form. Some of them will promote the formation of nucleosomes. Some of them will prevent the formation of nucleosomes. That is what determines whether a DNA molecule can be like this in some places, and some places it doesn't form so many nucleosomes. So here is a model that is shown here. Here there are two sequence specific, and these are the proteins that are binding, interacting with their respective sequences. because of their binding. In this location, nucleosomes will happen. In this location, nucleosomes will happen. But between you have two proteins, and for the formation of a nucleosome, we require at least 147, and if there is H1 also, it is about 167 base pairs. If it is less, if the space between, say, protein 1 and protein 2, DNA binding proteins, if it is less than about 150, then a nucleosome cannot be organized in this location. So that region will be left out, will be nucleosome free. But in certain other places, because a protein is bound, that will enhance the binding of formation of nucleosomes, something like this, right? As I was telling you about, so nucleosomes prefer bent DNA. So nucleosomes, when they form, they have to form a bent like this. If a DNA is like this and you want to bend it, it requires some energy to do it. What if there is a DNA that is already bent, like because of some sequences, it is bent like this? Then that is where it's much more easier to form a nucleosome. And usually that happens when there are regions of AT-rich regions within there, when the minor groove, say this is the region, so this is the major groove and this is the minor groove. When you have ATs, more and more ATs here, right, and then they tend to bend, and that is where that is favorable for formation of nucleosomes. Okay, if in this same place, if you have GC rich, then they do not form as much as base. Now say, okay, let me try it this way. I'll try to use linear DNA. Okay, that's not correct actually, but we will just use. If you have a lot of ATs, it will bend this way, say a little bit of bend, and the rest of the DNA is same way. If you have AT here, then DNA should be bending like this, right? Instead, think of it like this. You have a DNA, because of AT, it is bent in this direction. And you have GC here, right, which may have a little bit of opposite one. They might bend like this. And you have AT again, you might have bending like this. What is shown here is that. Okay, more AT regions will tend to bend this way, and the opposite effect is made by the GC ridge, because of which the DNA could be curved like this, and that is a much more energetically, much more and better DNA, or easier for formation of nucleosomes. So this is one critical point, critical part of the chapter where we are talking about histone modifications. That is a big part of epigenetics. In epigenetics, we will learn about it in the later part of the this course. But right now, you remember epigenetics as a level of gene regulation, right? Gene regulation, or alteration of gene expression because of covalent modifications, right? Covalent modifications. Covalent modifications can be in two types, at least two. The one best understood ones are about covalent modification of DNA and covalent modifications of histones. There are other modifications, epigenetic regulations that is done by RNA molecule. So DNA could be methylated. You don't have to remember that right now. What we are talking about right now is about histones. Remember the histone tails. This is a histone tail. This is a nucleosome, and DNA is wound around it. Say like this. Say for example, And we also discussed that the histone tails are extending outwards like this, right? How many histone tails would be extending? You would have 8 histone tails. Why 8? Because you have 8 histone molecules in the inner nucleosome. And these histone tails have many functions, including the formation of 30 nanometer fiber by interacting with the other histone tails. They also serve as sites where modifications can be done, right? On these histone tails, several covalent modifications can happen. The um, three important modifications that happen is acetylation, methylation, and phosphorylation. Acetylation and methylation of lysine and lysine residues, and the phosphorylation on serine residues on the histone tails. Some of the biggest histone tails that are very prominent that many people understand, we have better understanding, is H3. H3 is a histone. Some of the things that you need to remember is acetylation is usually associated with transcriptionally active regions. So if there is a part of the genome that is highly expressed in eukaryotes, all these histone, everything that we are learning is about eukaryotes. It doesn't apply to prokaryotes. They have a different thing, and they do not have something or not similar to histones. And if it is deacetylated, then they are usually repressed chromatin. Methylation can be associated with repressed or active chromatin. It depends on the position of methylation. Okay. And phosphorylation of H3 will make it highly condensed chromatin. When you say highly condensed chromatin, that means there is no gene expression, and especially when it is about metaphase of mitosis or meiosis, that is where you want all the chromosome to be to form very structures that are highly compact in nature for easy segregation. Then you need condensation to happen. That happens because of phosphorylation of H3. H3 histone. So here are some of the histone tails and what may happen in each of them. So here is the residue. This is phosphorylation, methylation, acetylation, and there is one more called as ubiquitination. If you read ubiquitination in cell biology, you would know that typically if it is polyubiquitination, it is involved with being as a signal for being degraded by the proteases. Monoubiquitination has a different function. But the majority of the histone tails that are the ones that are extending, why do you call it as S1 here? That means S is for serine, and that is the first position. Here is fourth, and here is fifth position. Lysine, lysine. K is for single letter code, lysine and lysine. Why are we counting from here? 1, 2, 3, 4, 5, and so on? Because proteins are always read from N terminus to C terminus. N is for amino group, C is for carboxylic group. So the tails that are extending out of the nucleosome are having the N terminus. So you start 1, 2, 3, 4, 5, and so on. So I hope we remember phosphorylation occurs on serine, right? Here is another a deviation from it, 309. Serine and 309 are hydroxyl amino acids. On the hydroxyl group, you have the phosphorylation. But in H3 is one that is very prominent. You can see that we understand a lot more about H3 than others at this stage. It has a very long tail, and it has a lot of hydroxyl amino acids and lysine and so on, because of which there is a possibility for a lot of modifications to happen. So in different cells, different residues could be covalently modified, and there can be a different consequence, which we will probably discuss in the last chapter, where we'll also have we'll discuss more about epigenetics. At least the brief introduction will be there. It will become much more relevant. For the moment, what I want you to remember is that there are histone tails, and there are serine and lysine residues, which can be covalently modified, phosphorylation on serine residues and threonine. Um, and then we have methylation, acetylation. Methylation and acetylation can happen on lysine residues. And here is arginine. R is for arginine here. So that is what you would want to remember for the moment. And there will be the type, which histone is modified, what is the modification, and at which position of the histone tail is modified, and how many modifications are there. All this cumulatively will determine what happens at that part of the genome. Right now, just remember about the modifications. So there are about, we will discuss with the figure that will help us, bromodomains and chromodomains. Okay, I was telling you a little bit about it in the previous slide. Bromodomains they bind to acetylated histone tails, right? And chromodomains, they can interact with methylated histone tails. Okay, so there will be many proteins which can interact with modified histone tails. And if they have to interact with the modified histone tails, they should either have a bromodomain or a chromodomain. Right? I will just put this as chromodomain. This part of it is the domain. This is one domain, and the other one is bromodomain here. Okay? So if there is any acylated DNA, acylated histone tails, then a protein will come which has a bromodomain, will interact with that. And this is an enzyme which could carry out any of the functions. Many diverse functions are there, which we will discuss later. Right? So you have protein A and protein B. What can protein A do? It can go and bind to histone tails that have an acetyl group. And then it binds using the bromodomain, it binds here and performs an activity. It could be activation of transcription and so on. If it were chromodomain, it will bind to methylated regions, and the chromodomain will bind here, and the enzyme part will carry out a function to it. So the domains that recognize acetylated histone tails are called as bromodomains, and the domains that recognize methylated histone tails are called as chromodomains. Here is a nice picture that will summarize a lot of things here. And let's see how we'll go. Here is the unmodified DNA. And all the histone tails have a lot of basic amino acids. Histones have about 20% of basic amino acids like lysine, arginine, and so on. So all the basic amino acids, they have this positive charge, and DNA has kind of negative charge. So they kind of tend to interact with the DNA and so on. If you have acetylated it, then there is a neutralization, or it's kind of negative charge added. And then what you will get is that the DNA becomes, the DNA becomes much more open. Not the DNA, sorry. The histone tails get much more extended out like this. That means they are much more open, right, for interaction with other proteins. And here there is an acetylated H4, and what binds to acetylated histone tails? that is any protein containing a bromodomain. That's the name given to that. They can recognize the acyl group. And then whatever else is attached to that protein. I mean, this is a one continuous protein. I hope you know what is a domain means. So a protein can consist of more than one domain. It is one single polypeptide. This part will be as one domain, and this is as another domain, right? So the domain that what is a domain to start with? Domain is a part of a protein that is required to carry out one specific action, interact. You can say it as interaction. That specific interaction here, it is binding to histone tails. So you call that as that the name given to that is called as bromodomain, right? There will be another domain which may carry out some function. That is the enzymatic part. Similarly, we may have methyl groups. Methyl groups do not have, see, they are just CH3, so they do not change much of the electrical charges on the histone tails. But they can methylate it, and then those are interacted by chromodomain binding or containing proteins, and then further activity will be made. Acetylation is usually, not always, but usually associated with expression. Why does that make it easier? Because the more these are extended out, they are more accessible to be interacted by other proteins, which might be involved in gene expression. So here is a whole list. I don't need you to memorize all these things, but just have a look at it, and there is no loss of it. But I think I would want to find out something here. Histone acetyl transferase complex. Those are the enzymes that actually modify the histones. You call them as HAT, histone acetyl transferases. Then you also have histone deacetylases. I think HDAC is what they call. That is called as histone deacetylases. Those are the proteins that add a group, acetyl group, or remove a histone group. Okay. And then there are also methyl transferases, histone methyl transferases, HMT, and you also have methyl demethylases, histone demethylases. Okay, these are the proteins that interact with histones and add either acetyl group or phosphor kinases will be the ones that protein kinases will be the ones that are adding the phosphates to the histone tails. Phosphorylase, for phosphorylation, you have kinases that add the phosphate group, or you have phosphorylase, protein phosphorylase, which removes the phosphate group. Okay. So what I want you to remember here is some of the acetyl transferases, histone acetyl transferases, they have bromodomain. Some have both bromodomain and chromodomain. Some of them have only bromodomain, and so on. And they particularly act on only one type of histone tail. They do not interact with all types of histone tails. How can they distinguish? It is based on amino acid residues that are present in the histone tails. So there are a large number of proteins complexes that act upon the histone tails, thereby changing the making the genome or the organization of chromosome much more dynamic. Some can be opened up for expression, some can be repressed or silenced completely, and that is how there is a large number of proteins that are involved in just maintaining the chromosome and opening up and maintaining the dynamicity of the chromosome. So how does a remodeling occur? Here is a 30 nanometer fiber, and there is a histone acetyl transferase, and there is a DNA binding protein here. Okay, so the DNA binding protein binds here, and then that slides open the binding of another protein, and because of which, say, once the histone acetyl transferase works on it, right, you have acetyl groups and the chromosome is much more opened up, expanded, onto which, because of which there will, because of opening up, you might have several proteins interacting with DNA sequence based on sequence specificity. And there you have a multi-protein complex called as nucleosome remodeling complex, which can then act upon, bind to the protein here, and then act upon the genome to sliding or exclusion of a nucleosome and so on. Thereby, what was actually a very compact genome, right, chromosome, part of the chromosome, is now opened up for interaction with several other proteins. And usually, when it is open up, it facilitates expression, right? When you want gene expression to happen, we want the DNA as open as possible. When we don't need that expression, it is better to stay in very closed or compact format, like shown in here. Okay, I think I will stop here. And there is in the last part of the video, you must have seen how nucleosomes can change in during replication. What happens, right? Because we are saying that DNA is something like this, and now we need to form two parts like this. That means not just DNA, you need to have also the nucleosomes. So what happens is the nucleosomes, the brightly colored one are the older ones, and the newly synthesized ones are shown in light colors. So as the DNA replication machinery is moving around in this way, the nucleosomes here are disassembled, and then some parts could be used up in either of the daughter DNA molecule. New tetramers and new H3H4 tetramers, new H2A, H2B dimers will keep on adding on to the newly being DNA that is being synthesized. That is how the nucleosome formation happens in eukaryotes. Okay. Yeah, there are some other proteins and many other things. We don't need to memorize all of it, but it is a good idea to watch a few. What is bromodomain? Bromodomain is something that binds to acetylated histone tails. What is a chromodomain? That binds to methylated histone tails. Okay, just remember that much. So these are some of the questions. I mean, I don't need you to, but I think you should know the actual, many times in examinations, they ask this for an essay question. Higher order chromatin structure, describe it. And when you are describing, you should be able to tell the components of it. You should also be able to tell the types of interactions and so on. What we'll do in the next class is we will do problems based on mitosis and meiosis. On Monday, I would want to use it for trying to solve a few problems that are likely to come. As I said earlier, I don't like to ask essay questions. Majority of the time during examination, you would be drawing figures with labeled figures, and you will be solving some problems that are associated with replication and cell division and so on. I forgot to ask a question. I think we will do it on Monday, right? Any questions?